I'm on. Am I on? Now I'm on. Hi, I'm Miss Sloan, and this is a review for APBio students. Everybody have the pear deck or wrote it down in case you have a friend coming in late. Okay, this is chapter 18, the origin and history of life. Here is a list of things you might want to review. Big ticket items. I talked about, can you support the claim that early Earth created conditions for life to evolve? That's assuming that you're familiar with all the different hypotheses, right? And I've got them all like summarized for you on single pages, and we'll go over those. And then I mentioned the flow of things like reducing environment, the chemical evolution that needed to occur, and then the biological evolution that needed to occur. I listed that off. And then I said, can you describe the role that extinctions play in the evolution of viruses? And then did you do your highly suggested reading and thinking? Just look it over. There's nothing there. You just look it over. I believe this is the one that has the tree of life in it. Okay, so it's easy peasy. All right. So the first one, origin of life, conditions on early Earth that made the origin of life possible. So this is the summary that we talked about before, and this was in the presentation. So just talking about the time, the reducing environment, that you understand the sequence, that there were inorganic gases there. I mean, it's the long and the short version of it is that the inorganic gases that were on the Earth, that if energy was provided, and let's review the energy that was probably on the early Earth, lightning, UV radiation, geological processes, and radiation from isotope breakdown, right? That energy you have there would be enough to have those inorganic molecules become organic molecules. And that was a hypothesis put forth by Operin. And then there was a man named who did an experiment. Who is the Miller did an experiment. Then once you have your organic, and there are other ideas there too, and we'll talk about that. I'm just painting the big picture. Once you have your organic molecules, your simple organic molecules like sugars and amino acids, then you want to build what out of them? Polymers. Polymers, when we think about the four important organic molecules, what are they? Proteins, okay. Carbohydrates, lipids, nucleic acids, right? We need all four of those in order to build cells, right? So you needed those polymers, and the big assumption is the same energy you had before, you have now. You have catalytic ability, whether it's at a hydrothermal vent or on the surfaces of clay, right? Or that it would spontaneously form. And that once you have those polymers, then you can start to build those early cells. What do we call an early cell? Protocell. And then, oh, broken shoulder boy, Paya, how you doing?
I'm good.
Are you in a lot of pain?
Yeah.
Are you high as a kite right now? Oh, sorry, he's not. That was a joke. Yay. What is it? Now, are you on some pain meds right now?
No, I'm not taking it.
Oh my goodness. Just
All.
all.
Like it's this hour.
So all soft tissue damage,
Yeah.
which is the hardest one to heal.
Yeah.
I'm sorry, baby. Clavicles intact. That would be this bone coming across here. Okay, good. Okay. I'm sorry. Okay. Don't make that boy take notes. Y'all are sharing with him, right?
Yeah.
You have some weak bio buddies. Okay. All right. Here we go. Okay, where was that? A protocell. But to be a true cell, what does it have to be able to do? Replicate. It needs genetic material and has to be able to replicate and pass that material down. And then we just go through the regular stages of evolution, right? The first cell, autotroph or heterotroph? Heterotroph. Prokaryotic, eukaryotic. Aerobic, anaerobic. Anaerobic. So it's anaerobic, right? It's a heterotroph and it's prokaryotic. And then you could later have autotrophs evolve, but were they cyclic or non-cyclic? Cyclic. And later non-cyclic, sorry. And then you have the proliferation of oxygen, at first absorbed by the seas, the water, right? And then eventually it starts to build up. And then that would kill off most life. And then you would have the evolution of what kind of respiration? Aerobic respiration. And then after that, you go from being prokaryotic to, and then after that, you go from being unicellular to, and then boom, we're starting to go on up to the Paleozoic era, and then we'll review those. Okay? Questions on the big tickets about that? It hurts just looking at you. That's got to be so painful. Okay, so anyway, that is all there. Also, I forgot to mention this about that oxygen, besides having the ability to do aerobic respiration, besides having that protective layer, ozone allowed organisms to crawl out of the water and come out on land. It would also help having an oxidizing environment with the weathering of rocks so that you have more soil, so you could have more plants. It's like a positive feedback loop, right? And more oxygen. All right, next, this diagram right here, let me walk you through it. I think for the most part you got it down, but you can see inorganic, and I listed them here. You don't have to memorize them. You just know that if you have any carbon, it's like carbon monoxide or carbon dioxide. It's not complex. It's not really organic. Then you have your simple organic molecules. What is this guy right here? Amino acid. How many amino acids do we have? Good. And then you hook these together to form your polymers. And all of this is chemical evolution, right? Up to and including a protocell. It's like you're a real cell if you've got some sort of genetic information, which we think is what? What was the genetic information? RNA. Good. RNA first world. And you're self-replicating, then you're a cell. And then what I tried to show over here is that you have prokaryotes and later you have eukaryotes. But notice it goes 2 to 4 here. I'm just saying these cells came before those cells, but all this has to happen, right? Heterotrophs, then autotrophs, cyclic, then non-cyclic, right before we end up getting to eukaryotes. Talked about the oxygen revolution and mass extinctions in unicellular before multicellular. Big picture over here, 4321, in billions of years ago, right? The Earth, 4.5 billion years ago. First cell about a billion years after that. Oxygen revolution, and then eukaryotes about 2 billion years ago, and then multicellularity about 1 billion years ago. Most of the diversity of life we talk about, like in the Paleozoic era, right? We talk about those not being in billions of years, but millions of years. So just keep those straight, okay? Keep that straight. Perfect. So on your hypotheses, okay, you have the primordial soup. You have that here. And then hypothesis by Oprah and supported by Miller. You have the Iron-Sulfur world. And then you have that extraterrestrial origin by meteorites. So I put a slide together. I don't know if it's next. Here it is right here. Okay. Showing you that organic soup. Then over here to the right, Miller's experiment. What's this picture up here talking about? Extraterrestrial. And then remember we talked about the hydrothermal vents, and that those could have been not only catalysts for monomers, but also catalysts for what? Polymers in the iron-sulfur world. So once we have our organic molecules, now we're ready for our polymers. And so then we have 3 here. We have the RNA first, because it acts as both the substrate and an enzyme. We have the protein first. Remember Fox and the proteinoid microsphere? So not only polymers, but also we can jump up to protocells with that, because when hot proteins cool, they form those spheres, right? So it's kind of like if they go with that theme, you take it a little bit farther as well. Okay, then the iron-sulfur world. And then I added a couple diagrams here to help you out. So just take a minute, read through the RNA first up here at the top. And then this one, it moves so slowly. That's why it's not supported. But here's the iron-sulfur world. You remember Fox. And then Karen Smith, that was that clay could work so that both the nucleic acids and the proteins evolved at the same time, with clay acting as a catalyst. So just take a minute and look through the RNA first timeline and the iron-sulfur world. See if that makes sense to you. Okay, so we talked about RNA coming first. Then we talked about that later DNA stored that info. Remember when we talked about, we look at the central dogma, DNA to RNA to proteins is how we know it now, but maybe it started as RNA and DNA was just a storage device, and then proteins worked off of that. Okay, on your iron-sulfur world, this is just what I mentioned in class. I just thought I'd give you a little more information on that, about the effect of those hydrothermal vents. But again, that would be your catalyst, but this moves very, very slow in that process. So that's not well supported as the RNA first hypothesis. Do you have questions you want to ask me about that? Okay. All right. Now, protocell, early cell, we talked about those liposome, those fat droplets, and that happen spontaneously. That can happen right now, you know. And the same thing with those proteinoid microspheres. So maybe there was a hybridization between those two. You have a lot of time here to make this happen. And you need some sort of barrier. So what's happening to the inside is different than what's happening to the outside. It's not just dissipating out away, but you can have concentration of those polymers and maybe their activities within that cell membrane as a barrier. And then when we talked about what kind of metabolism it has, for sure a heterotroph, because that's more simple. Want to go with the simplest explanation. Then everybody evolved as autotrophs, and then some lost the process to make their own foods. I don't see natural selection facilitating that. Do you see that? It's easier to consume nutrients, and that probably, I know that might be distracting. He's just got to clean the fish tank. I don't know what else to tell you, but I keep hearing it too. So it would be more likely that cellular respiration happened first, because you're just breaking down the organic molecules and releasing the energy out of it anaerobically, than even cyclic photophosphorylation. That would have happened probably due to competition. As the nutrients start to run scarce, those that can convert one molecule to another molecule and eventually use energy to facilitate that process, those would have been your first autotrophs, and then later non-cyclic. Okay, and then the evolution of the genetic code. We have talked about this as evidence for evolution. You see that same language repeated in all life for the most part, and natural selection would have worked on that as well. Okay, then we transitioned on day 2, and this part, well, does anybody have any questions they want to ask me about the first part? Okay, then day 2, just, I like this diagram for perspective, right? Now that you know about the Precambrian, the Paleozoic, Mesozoic, Cenozoic, when you see the formation of the Earth at midnight last night, and then we look, we're in the Precambrian, oldest fossils at 5 in the morning, first autotrophs here at 6 in the morning, but then we don't have non-cyclic photophosphorylation until much later. We have free oxygen. We don't have eukaryotic cells until 5 in the afternoon. We're still in the Precambrian. We are early multicellular, flat-bodied, worms, right? And then we're starting in the Paleozoic era between 9 and 10 at night. That's when we talk about the Paleozoic, the Mesozoic, and the Cenozoic. So those are all fairly recent in time. And man, like we said, came up the last 30 seconds of that 24-hour clock. Okay? So how do we know the timing on that? We talked about dating. There was relative dating and absolute dating. Relative dating, you're looking at the strata and you're assuming that the layers on the bottom are the oldest. And you can use things like what? What else can you use to help you with that? Index fossils that are only found during a certain time, like trilobites during the Paleozoic era. So you can use that as a marker to judge those layers before and the layers above. Absolute dating, you're looking at the rate of decay. Remember, we talked about what's going to decay faster, a Twinkie, Trader Joe bread, or Weber bread. Yeah, what? Trader Joe bread is going to degrade the fastest, right? So if we want to measure something, in the short term, we're going to use Trader Joe isotope. But if we have something that's really old, we're going to use the Twinkie isotope. And looking at that rate of decay, if an organism has a half-life of 50 years, okay, how much would be remaining after 150 years? So in 50 years, I would have one half-life, right? And then in 100 years, I'd have another half-life. So now I'm down to a fourth. And then another half-life to bring me to 150 years. So now I'm down to an eighth of that isotope relative to the current ratio today. Does that make sense? So by looking at the percentage of the isotope in that rock and knowing what it is today, I can tell how old something is. So I want to use an isotope if it's really old, right? When we look at our first fossil, and that it is 3.5 or 6 billion years old, we can't use carbon-14 to date that. That's the Weber-Bread, isotope. It's not going to work, right? We need something that breaks down a little bit slower, and we know that rate of decay, it's constant, and we look to see what percentage of that isotope is there. Okay, from that, in the Precambrian, the big ticket items are listed right here again. I've gone over those with you several times, but I wanted to include it again. And then other key things we want to remember, the importance of the oxygen revolution and how all the species that got extinct. And you can't see it because I'm blocking it with this, but we talked about multicellularity. But before we can talk about multicellularity, we have to talk about the evolution of the eukaryotic cell, right? And that's the endosymbiotic hypothesis. We have already talked about that. We're comfortable with it, right? Yes? You should revisit it if you're not. And then we can talk about multicellularity. And the irony of it all is it looks very similar to the development of an embryo, right? With the blastula and the gastrula stages. Yes? Okay. Then that brings us here to maybe baby. I'm afraid now to touch. So now we have our eras. So let's just kind of review our time in the era. So right now, the era we're in right now is and we go back how many? 66 million years ago? Million, right? These eras are in the millions. So 66 million years ago. So how are we going to remember that? Speed limit, and you're driving now, right? Then the era right before the Cenozoic era was and in the Mesozoic era, it ended when? 66 million years ago, because that's when the Cenozoic era started, right? Then what's the other? When did it start? So that's about when what? School's out, right? Okay. Then the era before that was the Paleozoic era, the old and Paleozoic. Okay. It ended when? Ended when? Right. And then we go back in time. When did it start? And that's about what time? Dinner time. And then everything before that is Precambrian, right? And in the Precambrian, that's when we're looking at Earth form, first fossil, all of that we just spent a whole bunch of time at the beginning. As far as what life we have, in the old and Paleozoic, plant-wise, where are we at plant-wise? Come on now. Algae. Boom shakalaka. And in the Paleozoic era, we go all the way from algae at the beginning of the Paleozoic era to the beginnings of gymnosperms. Those are your Christmas trees and Hanukkah bushes, right? And then animal-wise, where did we start? Invertebrates. So no spine, right? Flat, soft-bodied worms. And we go all the way up to the beginnings of reptiles, early reptiles. So there's a lot of transition there because we have to go from invertebrates to vertebrates, right? We need to go to fish and amphibians and then get ourselves to early reptiles. So that's a lot. That's why the very first period of the Paleozoic era is called the Cambrian explosion. Good. Mesozoic era is in the middle. Plant-wise, we have early gymnosperms all the way to early angiosperms, and animal-wise, we're going from early to early what? Mammals. Good. And then Cenozoic era is mammals, though we're not, fixed on humans, but we go mammals to humans, if you think about that. And plant-wise, we go from early angiosperms to herbs. Good. And then, that's about what you need to know. You don't need to know all of every single thing that evolved, as long as you understand the big picture from that. Okay. Next. is just the importance of extinctions. And we look at what kind of things could cause extinctions, whether it could be something extraterrestrial, whether it could be plates moving around and outgassing of volcanoes leading to global warming. But what we see, the trend, there are 5 major extinctions, but at the end of the Paleozoic era, it was known as the Great what? Dying. So many species wiped off the planet. But the pattern that we see there, when they get eliminated, they create vacancies and you get a new type of organism, right? So at the end of the Paleozoic era, so many animal species died, it now made room for all different kinds of reptiles to disperse. And then when you had your 2 extinctions in the Mesozoic era, then that made room when the dinosaurs became extinct. That gave room for mammals to start to spread. So that's what we want to see as a result of those extinctions. And then that is the end. Do you have any questions? Okay, yes. No, that's it. I would know there are 3 in the Paleozoic era and 2 in the Mesozoic, and we could be contributing to the 6th extinction. I would know that. Yes.
The highly suggested reading and thinking, do we just need to like know about the diagram or
Just
like
look at it and you're done.
okay.
Yeah. You're welcome.